that, let's welcome Chris.
All right, thanks, Jake. Thanks very much for inviting me. Always like coming out to Seattle. I'm from Vancouver originally, and so it's nice to be close to home for a little while. Like Jake said, I live in Nashville now, and I'm at the Vanderbilt University Medical Center, which recently spun off of Vanderbilt University, so I'm not actually a Vanderbilt University employee anymore. But at Vanderbilt, I do generally statistical computing in the context of Bayesian Inference, as Jake said, and also in decision analysis. And when you're a biostatistician, you have your foot in two worlds, one in the statistical world, one in the bio world. And my applications tend to be in the world of infectious disease, microbiome research, and evidence-based medicine. So today I'm going to talk about couldn't really decide what to talk about when I was invited. Do you want to hear about software stuff, or do you want to hear about applications? And so I'm going to talk about a little bit of both. So you'll learn what PyMC is, and you'll also learn what evidence-based medicine is. But so this is about the application in general. This is about the application of Bayesian methods to biostatistics, and it's facilitated by hopefully useful open source software. And the point of all of this is to make the application of statistical methods easier and more accessible, and that's particularly in the context of data science these days, where everybody wants to use data to drive their decisions, and so having software that's both open and capable is super important. So first, what is evidence-based medicine? You might be asking, what other kinds are there? We hope it all is evidence-based, but what we're talking about here is the use of modern best evidence in making decisions about the care of individual patients. And so there's always been this notion of clinical judgment and kind of the art of medicine associated with making decisions about individual patients, and that depended on the experience of the individual physician and their ability to determine what research is available and worthy of consideration. And so evidence-based medicine is an attempt to kind of formalize all of this and to create bodies of evidence, not to replace traditional applications of medicine, but to make it better. And you may not know it, but there's an entire agency in the United States that is partly devoted to this. This is the Agency for Healthcare Research and Quality, or AHRQ, as it's known. It's a division within the US Department of Health and Human Services, and it's there to enhance the quality and effectiveness of healthcare services. And what it does is it sponsors the generation of reports that are made to improve the quality of healthcare in the US. And it's generally via a bunch of centers that they sponsor called evidence-based practice centers. And one of them is at Vanderbilt. And what we do is generate reports, and these reports are based on science-based information typically on medical conditions or interventions, particularly things that are costly, and we want to know whether they work. What we do is we review all of the relevant scientific literature on a particular topic, on a wide spectrum of health services topics, and also produce technical reports on things like methodological topics and other types of evidence synthesis reports. And so what we're doing are systematic reviews, and these are very formal. You get meta-analyses from different places. They're not often full systematic reviews, and this is what we're talking about here. So there's a whole process that we go through. We have key informants, experts, subject matter experts that are brought in to establish typically key questions. What are we actually trying to answer here? What are the appropriate questions to answer? Refining those questions. We have a technical expert panel that's external to the process that gets consulted on these things, almost like an IRB. The review is posted on a public website for public review. And then from there, we develop a strategy for finding all of the available information. That's actually the hard part. Not only getting all the studies in hand, but then extracting the data and assessing whether or not any of it is any good or worthy of inclusion in the synthesis. And the ones that we've specialized in tend to be centered around women's health and pediatrics, because that's where our experts kind of reside. So these are ones I've been involved in. And you can see that a lot of them are having to do with, comparative effectiveness. So which pharmacologic interventions are good for infantile hemangioma, which interventions are good for helping women quit smoking when they're pregnant, those sorts of things. The last one is one I'm going to be talking about a little bit later today to kind of motivate the statistical stuff. But typically what we're doing is combining a whole bunch of studies, some of which are small, in order to get a better estimate of something that we're interested in, or a more comprehensive estimate, and see what works and when and where and with who. And the technical, sort of the quantitative angle of this, one component, this is sometimes the only component people see, is the meta-analysis that comes out of it, right? So we're talking about meta-analysis. These are the actual statistical methods that we use to integrate these studies that are related. They have to be related studies to produce some sort of combined estimate of something that we're interested in. And the conventional methods for this include things like estimating an effect size by combining a bunch of studies, estimating the heterogeneity. That's important too. If everybody's doing, if there's a large body of research, it can tend to be very heterogeneous if it's done over different populations in different places by different people with different methods. We can sometimes do things like meta-regression, where you actually take the effect sizes and regress them in a model. The nice thing it allows you to do if you have more data, more metadata, is to do subgroups analysis that you sometimes can't do in an individual study. Which subpopulations does a particular intervention work for? And then an important one, evaluating publication bias. Are we only publishing the things that work? Are we missing something? These are all pieces of it that can be approached using statistical methods. And the meta-analysis, like I said, some people only see the meta-analysis, but it really is the tip of the iceberg. And I didn't realize this till I started getting involved in an evidence-based practice center. So we get, we do reviews of abstracts. So first you get a bunch of abstracts, and if you do a search on something like, a recent one was autism spectrum disorder. Well, guess what? Hundreds of thousands of abstracts, and they all have to be reviewed, dual reviewed. Two people have to look at them. And then once you go through the abstract review, all of the studies get screened. You got to make sure that this research is being done on, not being done on horses, it's being done on humans, things like that will slip in occasionally. And then extracting the data. Did they actually publish the data? Or did they even publish all of their outcomes? And then we adjudicate the discrepancies. This takes months and months. Assessing the risk of bias, that's a very important component of this, is all the body research bias towards older men or something like that, or the methods themselves biased versus being the population being biased. And then just an overall, assess the overall strength of evidence. And that's before you get into doing the meta-analysis. Now, everywhere that meta-analysis, every field that meta-analysis has been introduced, there's been objections to it. There continue to be. And they come on a variety of fronts. There's suspicions of just the ability of being able to automate the integration of studies. It's hard enough to do one study right and get things kind of comparable within a study. How can you possibly do it across studies? Because there's varying qualities of studies. This notion of publication bias, do you even have all of the information at hand? And it's just, the major concern is usually everything's too different. You can't possibly combine these different studies done by different people in different places in the same framework. The problem is, though, is that in the absence of meta-analysis, people will still do this, right? You still synthesize a body of research. It's whether you do it informally, qualitatively, versus formally, quantitatively. And so attempts will be made to infer across the body of literature anyway. So you may as well be explicit about it. And as long as you keep in mind that it's just a model you're building, just like with any modeling approach, people either entirely trust it or they entirely distrust it. And kind of the same is true in the meta-analytic world. And this is kind of where some of the useful statistical methodology comes into play. Some of this stuff can be dealt with, some of these concerns can be dealt with using the appropriate statistical methodology. So in a traditional meta-analysis, one of the problems is kind of a default approach where you pool all of your studies into a single pot, essentially, all of the data or all of the outcomes. And you try to estimate a single quantity, theta, here. So we're pooling everything, essentially treating every study as being identical to one another. And you can see where you would might have an objection to doing that. And in the absence of meta-analysis, if you don't do this stuff, this is what you're doing. You essentially have an unpooled model where you say everything's too different, everything's got to have its own estimate, so every study stays in its own silo, and you can't really make any overall inference for this. Neither of these models is very good for doing useful things like making predictions about things that you're interested in, about making decisions. But there must be another way. There is another way. Hierarchical models is an approach to doing kind of halfway between those two extremes. What I've presented so far are two extremes to an approach, where the intermediate is this, where you say that a set of studies 1 through k are neither exactly the same, but nor are they entirely different, right? They're attacking the same problem. They're using similar methods. They're perhaps using similar populations. And so what we do instead is we model how different, we explicitly model how different the studies are across the body of literature. And we say that every study has this notional population from which they're drawn. And in some hospitals in some cities, you may get better results from an intervention because they've got a different population or they're doing things slightly differently. And in other cities, maybe you do poorly, and so on. So there's this population. You model this population. And you create a hierarchical model. So the hierarchy is you've got individual level or study level observations and then population level observations. And what this does is it partly pools your model. It allows you to share strength across studies without assuming they're exactly the same. Because studies are allowed to vary, but they're only allowed to vary within the constraints of the model that you've placed on them. And because of that, you're able to learn, because we'll be able to estimate these, what we call hyperparameters. So the hierarchical formulation kind of looks like this, where we've got a sampling distribution. So this is the information from individual studies. And we can say that this is study I, or individual I sampled from study J, but then there's a population distribution. So the bottom notation is we're sampling, say, effect sizes from different clinics or hospitals, for example. And this can be generalized a little bit, right? You can the hierarchical formulation only works if you can treat individual studies as being what we call exchangeable, that they're conditionally independent. Conditional on what? Well, conditional on maybe some covariates, some information that would cause them to be more different than you'd expect from some model. So how do we do this? Well, a natural way of fitting hierarchical models is using Bayesian inference, and you don't have to, but you'll see in a sec that it's very easy to accommodate hierarchical models using Bayesian notation because it's a probabilistic approach to modeling in general. Everything in a Bayesian model has to have a probability distribution associated with it, and what I've just related to you is implying some distribution of outcomes. One of the other things that's important in the context of meta-analysis is that it accommodates uncertainty in all aspects of the analysis. Everything has a distribution associated with it, either that describes the mechanism for it being different, or uncertainty in what we know about the value of some unknown parameter. So everything that is unknown, missing data, predictions, in addition to, say, parameters in a model, are treated as a variable with a distribution around it. And so what we do in Bayesian inference is calculate something called a posterior distribution. And this whole approach is known as inverse probability because we're inferring effects to causes. So we've observed some outcomes, some effects, and we're inferring back to the cause of them. So theta here are all the unknowns in our model, and y is everything we've observed. To Bayesians, there's only two things that are observed and things that are unobserved. Everything can be classified into one of those two things. And what we can do is use conditioning statements to try to determine what these causes might be. And so we're able to estimate all these unknown quantities. So these unknown quantities contain things we care about, things we don't care about, nuisance parameters as well. And again, the reason we do these, there's sort of philosophical reasons that some people use to approach Bayesian methods. I'm very pragmatic about them. My background is in biology and ecology. It's not in statistics. So I came about it because it helped me solve problems I couldn't solve otherwise. And it's very useful in this context because it's a simpler approach to statistics in a way, but that simplicity allows us to build actually more complicated models in sort of building blocks. This is the obligatory Bayes formula slide. This isn't a statistical methods talk, so I'm not going to dig in too deep into this. How many have used Bayesian methods before? Anybody? Oh, all right, keep going. Yeah, this is the only estimator you need to know for Bayes. I won't spend any time on it then. So, back to how it relates to hierarchical models. Well, the first step in Bayesian modeling is specifying a full probability model, and that's essentially what we're doing here. And then estimating the posterior distributions of all the unknown parameters. So here we're going to estimate the mu and the betas and the sigmas. Everything else are draws from this model. But how do we do it? So traditionally, what's kind of held Bayes back over the years is lack of our ability to calculate posterior distributions because they tend to involve intractable integrals. Can't do multidimensional integration very easily, and so it's important to have statistical methods, computationally intensive methods. Generally, that is what people have started to use. And there's a variety of software now available to do that. And so PyMC is one of those. I started this project back when I was a postdoc. Since then, it's grown to be a much larger open source project thanks to things like GitHub that lets you make your software available to a wider community. And if it's a good idea at all, people, smart people get attracted to it and become involved in it. And so what it's become is a very general probabilistic programming framework. And you can fit arbitrary models using PyMC. So you may be familiar with Stan or WinBugs, JAGS. It uses a similar but slightly different methodology, closer to Stan in a way. It implements what I would call next generation Bayesian inference methods that I'll describe in just a second. It's based on a framework called Theano, which is ostensibly designed to do deep learning. And we sort of hijacked it to do probabilistic programming Bayesian methods instead. For most of its existence, it was sort of a three-person operation, me and usually like 2 graduate students, and then they would graduate and I would never see them again, and then a couple more will come along. And then recently it's become a little bit bigger than that. We have over 100 contributors now, and we have about a dozen sort of permanent contributors to it. And it's being used quite extensively now, both in academia and industry. That's the paper that you can look at if you want to read about it, and the GitHub repository where it resides. When I talk about probabilistic programming, I'm referring to an approach, a programming approach that essentially defines new primitives. So we're used to building programs using variables that are integers or strings or floating point values. Well, what we're doing when we're talking about probabilistic programming is essentially manipulating variables that are stochastic objects that are able to generate random numbers. And this facilitates building probabilistic models. So we could have a variable that's a distribution over values. So something's normally distributed. You generate instances from that, variates from that. You can have more complex things like distributions over functions. So a Gaussian process. Every realization is a time series, for example. But the important thing that allows us to do is conditioning, where we can say that some binary outcome might have a Bernoulli distribution, a 0-1 distribution, and it's conditional on the value of some probability of an event that itself might be unknown. And what it does is it allows probability models to be specified at a very high level. And so however you specify the model, kind of Markov chain Monte Carlo is sort of been the workhorse of Bayesian inference over the years. Again, not going to get into too much detail, partly because you probably already know what this is. We're generating, it's not only is it impossible to analytically solve most Bayesian models, it's also usually impossible to sample independently from them. And so what we can do instead is sample dependently. So you can generate a Markov chain. And as long as that particular Markov chain satisfies the detailed balance equation, which is this relationship that implies reversibility, if you sample from it long enough, you'll get draws from the posterior distribution that's described by pi there. And so the workhorse of the most common subclass of MC/MC sampling is definitely the Metropolis algorithm, and it kind of goes like this, where you initialize parameters arbitrarily, and then you propose a new value, because we're not able to sample directly from it. You propose it, you evaluate the quality of that sample based on how it looks in the posterior distribution, and you either accept it, you add it to your bag of samples, or you revert to the current value and take another sample from your current value, or another copy of your current value. And that's what a lot of the packages like JAGs and bugs are based on, either Metropolis or Gibbs sampling generally, which is a subclass of this. And what you get is something that looks like this. So this is a simple, a big but simple model. What we're looking at here is sampling from a 1,000-dimensional multivariate normal. So a simple model, but a big one. And you're looking at the marginal posterior samples from 2 of those. And what you can see is that it's not doing very well. So it's being sort of stuck in, not quite the tails of the distribution, but you're definitely a biased part of the support. And you can also see that it's kind of jerkily sampling and then stopping sampling, and because a lot of these samples are being rejected. Metropolis sampling, though very easy to implement, typically for models that are of even reasonable complexity, tend to require tens or hundreds of thousands of samples to converge. Sometimes they just never converge for relatively easy models. It's not very efficient. Only about 24% of its samples are accepted when you're behaving optimally. And so, again, sort of a newer reason for people not using Bayesian methods is sometimes that it just doesn't work. The methods sometimes just don't work with your models. And so there's been a newer class of MC/MC algorithms that do a better job than just a random walk. What we saw before was just a random walk that sometimes works and sometimes doesn't. But we can do a little bit better than that. And in particular, a class of algorithms called Hamiltonian Monte Carlo algorithms can be applied. And Hamiltonian Monte Carlo uses invokes an auxiliary variable and uses gradient information about the model. So what it does is it emulates Hamiltonian dynamics, physical dynamics, on a Euclidean manifold. And so it breaks your problem down into having a position and a momentum. So your actual point is a position on a landscape, the landscape being your posterior distribution, and then it moves along with a particular momentum. And so we define these two quantities and simulate how those two quantities change over time based on the gradient in the model. So we're not doing random walks anymore. We're using gradient information to inform where proposed values are. So calculating gradients is important. And what happens, the algorithm kind of looks like this. Because we're simulating using a computer, we can't simulate continuous dynamics very easily, so we have to discretize it. So what happens is we simulate a bunch of discrete steps alternating between changing the momentum and changing the position until it obtains a new proposed point. And what it's doing here is it's exploring the level set, essentially, of the energy of a piece of the distribution. And then it samples a new velocity. So essentially, you took a marble and you put it in like a salad bowl and you roll it along, and it will just keep going like this, right? And Eventually you stop it, and then you flick the marble again in a different direction, so you sample a different level set of that distribution. So we have this velocity variable that is usually just univariate Gaussian. You perform a bunch of these leapfrog steps until you stop, and then you do because we're doing this discrete approximation to a continuous system, you actually still have to do a Metropolis accept reject. If we were able to do continuous simulation, you wouldn't have to do that. So each step can be quite computationally expensive, but the samples that you get end up being a lot better. So here's the same problem with Hamiltonian Monte Carlo, and this is a relatively easy problem for it. You can sample on a torus this way. You can, crazy Rosenbrock functions or things like that. You can create almost arbitrarily complex problems, and it's able to do that as long as you've got this gradient information. And that's where Theano comes into play. PyMC2 was essentially a Fortran library glued together with Python. and PyMC3 converts that computational backend over to Theano. And the important thing that Theano does is automatic gradients. So here we're defining a matrix and a transformation of that matrix, and then the fourth line there, automatic differentiation. That's really the hard part about doing Hamiltonian Monte Carlo, is for arbitrary models being able to take the gradient. And we're blessed now with a ton of frameworks for doing this. We've got TensorFlow, we've got MXNet, we've got Torch, and all of these things. So it allows us to do this efficient automatic differentiation. It also allows us to run this on GPUs and so forth. So it's really great. And the front end on it is really easy. We just build a specify model. I'll show you a little bit of that in a second. And then we just run sample on it. And it picks an appropriate specific HMC algorithm. This one's called NUTS, the NUTS sampler, no U-turn sampler. Andrew Gelman was just here. He probably mentioned NUTS, right? No? Yes? And it auto-tunes itself a little bit. And so we just draw 1,000 samples from that, and we're good to go. The other newer method for doing Bayesian inference takes a different approach than a sampling algorithm. The problem is MCMC, even using an enlightened approach like Hamiltonian Monte Carlo, can still be very slow. It's very computationally intensive. It does not scale well to larger data sets. It scales very poorly with it. It scales very well with the size of your model, but not so well with the data. And of course, we live, in the age of big data. We still want to use Bayesian methods. And so another approach is to do an approximation using another distribution. This is what variational inference does. So we take a simpler known distribution, and we optimize it. We change its parameters, optimize its parameters such that it's as close as possible to the true posterior distribution. And what do we mean by as close as possible? Well, we use a measure of divergence called Kullback-Leibler divergence. It's an information theoretic measure of distance. And so we try to minimize that divergence. The problem is this is the form for Kullback-Leibler divergence. The problem is that the expression for it involves the posterior itself, which we don't know. We can't do that. But we can re-express it in terms of a quantity called the evidence lower bound, which only involves the joint distribution of the data and the parameters, which we are able to access. And then it'll have a constant at the end. It's only a function of the data. It turns out that minimizing the Kullback-Leibler divergence is equivalent to maximizing this lower bound. And so this algorithm is actually kind of like an expectation maximization algorithm. The main issue with it is that it requires a lot of decisions on the part of the user. Well, which distribution do you pick to approximate it? And how do you do that generally? The problem is how to do that generally in a software package like PyMC, where you can throw potentially any model at it. the big innovation that allowed this to be applied more generally is an automated version of variational inference. Alp Kusekelbier and company, including Andrew Gelman, developed automatic differentiation variational inference. And what it does is it uses transformation. So it essentially transforms your model, no matter what it is, to the real coordinate space and standardizes it, and then just puts normal approximating distributions on it. And this works really, really well. And PyMC3 implements this. And so what ends up happening is now you get a trace, but these aren't samples. This is the ELBO as you run a certain number of iterations. And it doesn't usually take very long. Operates much faster, scales much better with larger data sets. What it asymptotes to depends. Your mileage may vary. Depends on what you used to approximate it. But it can sometimes get you a good enough approximation in a much shorter time than using MC sampling. And so similarly in PyMC, we can, rather than call sample, we can call fit. And so fit is just running, in this case, full rank ADVI. It's using a full rank matrix, covariance matrix. And you can see it runs, what did it do there? 200,000 in a few seconds. And then you end up with this approximation object, which is an approximated posterior, but it's a known form, and so we can sample from it. So we can actually generate samples that look like they came from MC. In terms of building models in PyMC3, I'm not going to get into too many of the details other than to show you the general structure. This is a very simple hierarchical model, meta-analysis model. These are learning outcomes from schools, from 8 schools. And what we do is we use some nice Python idioms, in particular here something called a context manager, the with statement. And what it does is it essentially creates a model, opens it up, and allows you to add variables to it. So any variable that's declared inside that block gets added to the model. And what's happening in the background is that we're building this Theano graph. So we're building a static graph that gets compiled and optimized. And then we run HMC on that. Because building Bayesian models is all about constructing a directed acyclic graph and then fitting your model over it. And so all of these, and you can see now now that everybody's running Python 3, right, we get Unicode characters. So we get to use pi and mu and sigma and things like that. So we get a prior on the mean, a prior on the standard deviation, and then here's the hierarchical model, right? So the thetas are the individual school results, and they come from this population. And then our likelihood is just another normal distribution, but we tag it with another argument here that's the observed data, and that says that it's a likelihood rather than a prior or a model. So 4 lines, you can do a meta-analysis. It's pretty nice. So back to evidence-based medicine. Here's kind of a real example from some work we did a few years ago. This was an AHRQ-sponsored meta-analysis or systematic review for estimating the probability of IQ impairment from PKU patients. Phenylketonuria is a metabolic disorder where children are unable to, their bodies are unable to metabolize the amino acid phenylalanine, and it builds up in the bloodstream, and it results in intellectual impairment. So low IQ, delayed speech, behavioral abnormalities, and seizures. And you typically get diagnosed at birth. If you've had a baby, you might notice they get sorry, you can't really see it in the background here, but that's a little baby's foot being pricked. And they take a little blood sample and they test for PKU. So it's typically screened early. And if it's not treated, and kids have chronically high phenylalanine levels, that's when the intellectual impairment starts to happen. And so we want to know how high does that have to be? What are there threshold levels of phenylalanine that tend to lead to higher probability of poor outcomes? And so the key question that was established from the initial project scoping and screening is what is the evidence that any specific phi levels are optimal for minimizing and avoiding cognitive impairment. And we're going to call cognitive impairment 85, IQ of less than 85, which is a standard deviation lower than the mean, population mean. In our data set, meta-analyses are very rarely big data sets. So we're talking about 21 papers that came from 17 different studies, mostly in Europe and the US. Some of the studies, so these tend to be small studies. PICU is a very rare disease, and so there tends not to be much money invested in its research. And so this is a key area where you want to use meta-analysis, where you can take a whole bunch of small studies and potentially improve your inference by jointly analyzing them. And the important covariates here were two things. One was whether or not the measurements of PK of phenylalanine took place in the critical period. So the key formative interval is kids that are less than 6 years old. That's kind of when the impairment will happen, if it's going to happen. And then whether or not the measurements were taken concurrently with the study or historically. So relative to when the study took place, whether the measurements were historical or not. So whether there's an offset. So some were concurrent and critical, some were concurrent and non-critical, and et cetera. We fit two different meta-analytic models, so there is a point at which you can't really combine everything into one. Ones that were more than 12 months before the IQ measurement were treated as historical. Ones that were concurrent were measured within 6 weeks prior to IQ measurement. There wasn't anything really in between, as it turned out. And the group decided that it was really unreasonable to combine measurements in the same model, given the potential for very drastically different relationships between those two variables, depending on how far apart they were taken. So we have a historical model and a concurrent model. And so the model looks like this. So we had assumed a linear relationship between expected IQ and phenylalanine. And what we see here is, oh, no pointer. We allow for the intercept and the slope to vary by study. So the slope is hierarchical and the intercept is hierarchical. And this random effect is included to account for a whole bunch of potential sources of variation in IQ, such as the IQ measurement, the measurement scale itself. There are lots of different IQ measurement scales, geographic location, any number of things that are unmeasured. And that's kind of what's going on into this random effect distribution. And then the slope included a fixed effect. So the third line here is a fixed effect that included the coefficient having to do with whether that study, the phi measurements were taken during the critical period or not. And then another nice thing that we were able to do is, most of the time you read a paper, they don't give you the raw data, right, that went into their study. Most of the time they don't. Hopefully this is changing as sort of reproducible research methods become more common. But often you get only summarized data. So what was the mean IQ and standard deviation? What was the mean phenylalanine of this group of patients? And that's only partial information, but it's nice to have. And so we were able to also roll in summarized data. 12 of them, in fact, only had summarized data. And so it turns out that the hyperbolic function can be used to transform a correlation, which we can get from summarized data, into a normally distributed random variable based on the Pearson correlation from a particular study. And the standard error is simply a function of the corresponding sample size. So for some of these, we could get expected values of slopes for the relationship. And so our directed acyclic graph looks like this. So it includes individual paper data, individual study data, summarized study data, with a couple of random effects, one for the phenylalanine effect, then IQ. And we incorporated empirical sampling variances, and then coefficients for things like the critical period that I talked about before. Another thing that Bayesian methods make really easy is imputation of missing data. You can do things like mean value imputation, which sort of glosses over the uncertainty in that imputed value. The nice thing about missing values in a Bayesian context is, like anything else that's unknown, you treat it as a random variable and you estimate it. And so in PyMC3, the code here is a little small, but a nice feature of it is that if you pass a likelihood, a NumPy array that is masked, so they have a particular class of NumPy arrays called masked arrays, where the masked values are the missing values, it will create a hybrid node in the model that will treat the non-missing values as data and the missing values as variables, and you'll actually get distributions for all those missing values. So we used a gamma distribution to estimate the distribution of phenylalanine values, and then it will sample from that for each of those. It does make an assumption about the type of missingness. This is assuming a missing completely at random model, which isn't always true, but it's better than putting mean value imputation into that, and we are at least accounting for some sort of uncertainty in that value. So now we have a whole model, missing values and hierarchical random effects and so on. We run it, and it breaks. It doesn't work. And we try really hard to give users the problem with black boxing something like this and saying, oh, look, you can do MC just by calling sample, is that you sometimes don't know whether it works or not. And by works, it means not just does it break and cause an exception, but are the values valid and reliable? And what we get here, let's see, the acceptance probability in chain 0 does not match the target. Chain 0 contains 242 diverging samples. And we can get diagnostics plots like this. These are marginal energy and transition energy distributions. When things go well, these two distributions, one is the marginal energy distribution of the entire Hamiltonian Monte Carlo system, and the others are the energy distribution induced by the momentum resampling, so that initial resampling. They should be similar to one another. And what that means is that there's a good chance that the posterior distribution is being fully explored, that you're not just getting a biased subsample of the posterior distribution. And we're clearly not here. So this is bad. So these are some diagnostics. Turns out one of the problems that you run into when you're building hierarchical models is they can be very hard to gain true convergence. And the problem is in our parameterization of the model. And this is normally what we would do. We would say that our population of, in this case it was the slopes of the study, are normally distributed, and we give it a mean and a standard deviation, and we add that random effect. And the problem is that it induces some pathological behavior in the sampling algorithm. So what we see here, so what happens is that when the group variance, what we have here is the variance of this group, and then a slope sampled from just one candidate slope. You could do them for any, they'll all look the same. You get this sort of funnel shape because, of course, when the variance of the random effect goes down, the individual values will get closer and closer together, right? So it'll be wider at the top, narrower at the bottom. So it's a funnel-shaped relationship. And in itself, this isn't the problem. This is what we expect. But the problem is that if the sampler is tuned for the wider portion of this, it's going to do very poorly when things are narrower. And so the neighborhood close to the lower bound of sigma is sampled very poorly, and that's going to result in bias. And you can see it in the trace here. So here is the sigma, and you can see right down when it gets closer to 0, it kind of gets stuck for a while. And then these are all of the slopes, and you can see they get squeezed, and then they come back out again. And your Gelman-Rubin diagnostics and Gavecki diagnostics, they're not going to catch this at all. So this will pass those tests usually. Turns out it's actually pretty easy to fix. You use a different parameterization. You reparameterize it. So this is a nonsense. So the last ones, the random effects were centered on that mean, mu. What we can do is reparameterize our model such that the random deviates are no longer explicitly modeled as being centered. So what we do is we have a mean and a standard deviation, and then we just have standard normals that we rescale. We scale it, and then we location transform it by the mean. And then, ta-da, it runs. And now it can sample the entire distribution. Our diagnostic plot looks better. We don't get any warnings. And importantly, the funnel plot now, the tornado has now touched down in Oklahoma. And you can see that now we don't get the trace being stuck in areas of small values of sigma. And these have real consequences, right? So these are the two distributions, posterior distributions of sigma. And you can see that the bottom end of the top one is completely cut off. And so you end up with biased inference here. And again, standard diagnostics aren't going to catch this stuff. Anyway, so these are things to keep aware of. And these are things in designing the software that we try to have warnings, at least, to kind of keep users aware of, because most people aren't usually aware of this behavior, among other things. And so in terms of how did it all turn out, so what we have here now are estimates of the probability. So how do we address that key question? Well, we have threshold values for IQ that we're dealing with, and we're looking for threshold values of phenylalanine. What you don't want to do is go in and discretize your data and say above 85, below 85, or phenylalanine above or below a particular threshold. You always want to use you never want to discretize continuous data. What we did instead was run the model on the full continuous data and then infer at different levels along both of those thresholds. So the dark lines here are using a threshold of 85. The lighter lines are using a threshold of 70, which is 2 standard deviations below population IQ. And the points on the line correspond to the integral of the posterior predictive distribution of the IQ up to a score of 85. So this actually, you don't see error bars on this, but it includes the uncertainty because we've integrated over that posterior distribution. So these are probabilities of dropping below 85. So now this can be used as a decision support tool, right? You can decide, is there a critical level of phi where there certainly isn't a step function of any kind, but you can kind of let clinicians decide what's high, or patients too. What is a appreciable risk? It's really hard to keep your phi if you've got PKU below a certain level. You've got to avoid protein. It's hard. It's very hard. And so you want to know how much you can have. You can't avoid all protein. And so what's an appreciable risk? If it's around 1,000, it looks like it drops. You're looking at about a 30% risk. If you double the phi, it goes all the way up to 80%. And so this can be readily adopted as a decision support tool, and it consists of a range of studies that each of which dealt with different parts of this line, right? So it's cobbled together from several studies and not just one. And then this is the concurrent measurement model. So, and as was predicted, the historical measurement is much more responsive. When you're older, it doesn't matter as much. The cognitive impairment is usually either happened or it hasn't, and so you get a relatively flat line down here. So, kind of the take home, to wrap things up here. Hopefully the open source tools of things like PyMC and Stan. Oh, look at that. Are make fitting reliable models hierarchical models for meta-analysis and other things relatively easy and accessible for a wider audience, people that weren't able to access these things before. And the nice thing about it is that it closes the lag between the development of new statistical methods and their application and uptake by users, particularly these variational inference methods. Some of the methods that we've got in PyMC, like normalizing flows, and some of the newer VI methods, some of them are still just archive papers, or they've only recently showed up in peer-reviewed literature. And so this is really great, allowing people to use these better and more widely. And we hope, in terms of meta-analysis, the hierarchical model somewhat reduces these outstanding objections to meta-analysis in terms of, again, not treating everything as either being exactly the same or entirely different, and also very explicitly describing the assumptions that you're making in running that meta-analysis. This is, just want to thank the current PyMC3 team, which is, this list is getting nice and long. I think the investment of time to support open source software project really pays dividends, and half this list are work in the private sector. They're not academics, and a lot of them are supported explicitly to work on PyMC from their employers, because there's, I think, becoming an appreciation of the value of these open source products on the bottom lines of large companies. And so